Thanks, Joe. Thanks everybody for coming. You're in for a treat tonight. I first met Elaine a few years ago. I went up to Galiano Island for an event that IMERSS was holding in their early days. IMERSS is Institute for Multidisciplinary Ecological Research of the Salish Sea. And it's a very Vashon Nature Center-like nonprofit up on Galiano Island. And Elaine is one of the advisors for that group. And within 5 minutes of meeting her, I was just spellbound with her stories about this microscope. So, and I was able to see some of the pictures she showed me. And it's pretty spectacular what you can see with this thing. So I'm so grateful, Elaine, for having you join us today here. Elaine is from the University of Victoria's Advanced Microscopy facility. So she works with this microscope, SEM. SEM even joined on its own to this Zoom. So it has its own little membership here in our circle. So, so Elaine is going to show us around with that. She's got a few slides that she's going to show us in the beginning. And all of us here, being islanders, are familiar with eelgrass because it's in many places along our beaches. The small thin blades and of the Japanese eelgrass and the thicker blades of our native eelgrass create these amazing nurseries of life for us. And they're really good habitats for juvenile salmon to forage in and forage fish. You can find those beautiful nudibranchs and all sorts of sea stars before sea star wasting disease. Anyway, we had more of them, and tons of critters in a lot of these eelgrass beds around the island. And so Elaine is going to take us even deeper and smaller into the eelgrass beds today to see things that we couldn't even see if we walked through and looked at these with our naked eyes. So with that, I'm going, I should actually go through a little bit. So what we'll do is we're going to have Elaine sharing her screen in a little bit. And once she does that, you can adjust your settings if you want, so that you're only seeing a speaker window and the screen. And it's different for different computers. So let us know if you're having trouble maybe in the chat and we can help you with it. So we'll be looking at Elaine's screen, and then we want this to be interactive. So Elaine's going to be asking us questions, and if everybody is familiar with Zoom, in the toolbar at the bottom, you can see a little place that says chat. And if you click on that, you can type a question that you have, or you can type an answer to the question that Elaine has asked, and I will be looking at the chat as we go through, and I will be able to call on you or ask some of your questions for you depending on the flow. So that's how we're going to do it for tonight. And you can also put other technical questions in there, and maybe Joe can help you with those if needed. Okay, so Elaine, welcome. And thank you so much for coming tonight, and go ahead and share your screen and we'll get started.
Okay, so before, well, no, let's just share the screen straight away. So, we have a sailboat that we sail in the Salish Sea, and this is off Sydney. And Sydney Island has a lot of eelgrass around it. And there was a piece of eelgrass just floating by, and I picked it up and put it on the deck and dried it down. Now, I'm sure then you're familiar with this kind of thing, but as it gets older, it gets to be more browny looking. And that's because as you look into it, it's got stuff growing on it. Oh, shoot, I opened up the wrong one, but that's okay. No, it was the wrong one, but we won't worry about it. That was just introducing the piece of eelgrass. So I'll stop sharing there. Oh, did you see that? Remember, sorry, I can get so, I'm just going to open this for you because Bianca and I were talking a bit earlier about this BioBlitz thing. And if this thing opens up, oh, it's taking too long. Oh, shoot. Sorry, Bianca.
No problem.
The problem with being an electron microscopist is you get sidetracked very easily. And
There it is. We can see it.
So this turns out to be a fern spore, but I don't know which fern it is. So I sent it off to my fern expert who didn't know. So she sent it off to her fern expert who didn't know. He thought it was a maidenhair fern. It's not a sword fern, because the stub that this was on had a sword fern on it, and the spores are quite different. But this one came in with a pansy petal. So I've now got to go into my garden where the pansies are and search a bit more for the fern. But isn't that beautiful? Anyway, see what I mean about, so I'm going to stop sharing there. And now I'm going to share the SCM screen. So you can all see the SCM screen, and I hope you can all hear me. Maybe I should stop sharing there so that the screen that you see is the big screen, so I can show you this tabletop scanning electron microscope. So Hitachi gave me this microscope for school programs, and it goes into school. And I took it out to Galliano for a community event. And one of the people brought me in some seaweed. And we just, this is such a good, easy microscope. You just put the sample on the stove and stick it in, and you can look at it. And there were these diatoms on the seaweed. So it kind of gave us this idea that seaweeds of all kinds can carry all kinds of other things. Now that COVID struck, of course, we don't go into schools anymore at the moment, but the schools are coming to me by Zoom. Now, if I turn my computer around, and you can see over here, this one here at the back, whoops, that one that I'm pointing at is, oh, it's only about $2 million. This one here is behind me, the one we're using, is only $100,000. But that's Canadian, so yeah. And so the one down the corridor, well, it started off at $8 million, but a whole bunch of other stuff got put into it. But it looks at atoms. But you've seen one atom, you've seen them all. In fact, I go to the bank and the bank teller says to me, I know all about atoms, he says. Never trust an atom. They make up everything. But that's the bank teller, right? So let's go into sharing the screen. Okay, so we have this. Okay, and I'll just close that up. All right, so it says ready for observation, which means that I've got a nice vacuum in here. And there's a start button at the top left, and we click on the start button, and we wait for the electrons to come down the column. Now the electrons hit your specimen and kick out secondary electrons. And the secondary electrons are picked up by the secondary electron detector. The detector doesn't move, and the specimen doesn't move. It's the beam that moves across in a raster fashion, hence the name Scanning Electron Microscope. So this is the top end of the eelgrass. And it's a cut, we've just cut off the top end. Now I have to go to fast to be able to move in real time. And if I just zoom in, let's say I zoom in to say 1,000 times, and it's all out of focus, right? So all we have to do is hit the auto focus button and the auto brightness contrast button, and then we can look at it in the screen. So diatoms are single cells. If you go to the ocean and you see a greeny, orangey, browny bloom, it's usually diatoms. And these ones, well, we ask the kids, what do they remind you of? And the kids say cocoa beans, which is great because the scientific name of this group is Coconeus. And you're going to hear Coconeus a lot, but it's a single cell with a silica coat, and silica is what we use to make glass. So this is a plant that lives in its own glass house. And there are a few other species here, in between these coconeas. But if I take this down, I have to go back into it. There are a few other animals on here, on this top end. This microscope is controlled by two knobs on the front, the stage, whereas everything else is controlled by a mouse. Now, most of the kids know how to use a computer mouse, but all the young ones don't. The 6-year-olds don't know how to use a mouse. They are touch screen people. But they learn very fast that when you've got a mouse and you use the wheel and you zoom in, you can see. So zooplankton are the animal planktons, and this guy is one of the most abundant. He's in a group called copepods. If they have very long antennae, then they're herbivores. If they have short antennae like this one, these antennae here, then they're usually predators. And so the school kids labeled this one Lewis, but I'm going to take you to see Bob in a minute. But there's another animal on here. Where is he? Over here somewhere. There he is. He's a filter feeder. And so he kind of looks a bit like a deflated balloon or a hot water bottle. And if we look in his mouth, I got to put his mouth in the middle so that when I zoom in, we're zooming into the middle, we can see what he's been feeding on. Now he's out of focus, so I'm going to take the reduced window and I'm going to drag it to where I want it to focus, because I want it to focus inside the mouth. So we do autofocus. Now, 30 years ago, an SEM used to look like the inside of a jumbo jet cockpit, but nowadays it's computer controlled. And the more sophisticated the microscope, the easier it is to use. So you can see it's been feeding on little tiny coconeas. So it's an animal that lives well with lots of food around it. There's also the matchstick kinds. There's just a few matchstick kinds here. And then there's this one over here. I'll put him in the middle. And we usually say to the kids, what does it remind you of? And I had Christmas cracker, and I had marshmallows on a string, and I had Kinder Surprises. But Kinder Surprises aren't being sold in the States anymore because they've got things in them that the young kids can choke on. So that's the top end. If we move down through to the medial end, the middle of it, we find, this is the stub. We find another piece that I've got here. And now we find we've got a lot more. There's a lot more coming in on this piece. And it's out of focus. So we click on the autofocus button. And we click on the auto brightness contrast, and we do a slow scan. So now we've got a lot more of the matchstick kinds. We've got a few more coconeas and a whole bunch of more marshmallows, which sometimes think of as necklaces. And there's two tintinids side by side. And here is an annelid. So if I go back to fast and I move in, if any of you have looked at an earthworm, you know an earthworm is segmented, but it has holes along its segments. And this one is a marine annelid. And if I go, it's out of focus, so I'll go autofocus. And if I do slow scan, you can see it's got little holes along the segments. And these are nephridia, which are kidneys. And you can store fats and you can store carbohydrates, but you can't store proteins. And proteins are broken down in the body to amino acids and glucose. Well, the the amino acids are broken down into ammonia and glucose. And the ammonia is toxic and it builds up very quickly. If you live in water, like in a lake or in seawater, there's all this water around you, and ammonia is very soluble. So you just pass it through all membranes of your body. If you were a chick in an egg, you don't have a lot of water around you. So birds make it into a solid, uric acid, and they leave it behind in the eggshell. And then when they're flying overhead as an adult, they still keep the uric acid. So you get a plop on your car and not a whoosh. But in the seawater, the kidneys are really just to keep the salts in the body at the right concentration. These holes here, you can just about make out the segments on it. Now I'm going to take you to my favorite ones. So if we move over now to another piece over here. They fell off onto the stub. They fell off the eelgrass onto the stub. So here they are. And you can see that they're triangles. So these triangles have a scientific name of trigonium, amazingly enough. But the kids call them Toblerones. So these are the Toblerone Diatoms. And if we zoom in here, and we do a focus and a brightness contrast and a slow scan, we'll up the contrast. And we'll down the brightness. You can see that the holes have holes within them, and there's this pattern. And you can tell one species of diatom from another by the hole pattern. But if you were an artist or even a doodler, I don't know, I think that pattern would be very difficult to put together. But it's something a doodler might do. So diatoms are having this hard outer shell, need the holes to let things in and out. Diatoms, though, keep us alive. And so they make 20 to 50% of the world's oxygen. So a tree is important because an average tree can keep 4 men alive for the year. But diatoms, when they, in the spring and the fall, when they have a bloom, they provide 50% of the world's oxygen. So they're only tiny, but they're really, really important to us. But on this diatom, you can see some gunk, and that is broken up pieces of shell, of diatoms, and it's called diatomaceous earth. And we use diatomaceous earth in our everyday lives. If you make beer, then you use it to filter the beer. Vancouver Aquarium uses it by the ton to filter seawater. Gardeners use it on plants to keep the insects off because it's little pieces of glass, tiny little pieces of glass that tiny insects don't like eating. And it does protect your plants to a large extent. First Nations used it to clean blankets and to protect the blankets from insects as well. So I do a lot of work with First Nations blankets, Coast Salish wool dog blankets, and I find that we often find diatomaceous earth in the blankets. If you add nitroglycerine to diatomaceous earth, you have dynamite. So it can be used in all kinds of ways. Okay.
Elaine,
Yes.
those are so beautiful and the patterns are incredible. I noticed that you were talking about that they have these, you know, the little holes, and it looks like it looks like they have smaller ones around the edges. Do you know if there's any reason for that? Like why the pattern changes there?
when one of the kids asks me a question like that, I'll say, maybe you'll be the scientist to find out. And that is our standard answer when we don't know.
I can see why these are your favorites. They're beautiful.
Okay, so let's go have a look at another animal on this thing. And you can see it's too contrasty, but by now you'll have recognized that we're going to do auto brightness and contrast, and we're going to do an autofocus and a slow scan. So this little guy here is a baby amphipod. So if you go to the beach and you pick up a rock and you see all these sand fleas skittering around and jumping about, they're usually amphipods. Amphi means different and pod means legs. So this is an animal with lots of different legs. If you go through the forest and you pick up some dead wood, as you lift it up, you probably find some wood bugs under it. And they're isopods. Iso means the same and pod means legs. So they are flattened like a dinner plate and all the legs are the same. These guys are more flattened like a book. All the legs are different and they usually swim on their side. So with this guy, oh, let me take him down a wee bit. So these are the antennae. You can see my cursor, yeah. And there's the head and there's the back and there's the legs sticking out. And there's claw legs and all kinds of legs. I'm going to put this bit in the middle. And we're going to have a look. On his back, you can see he's got a lot of cochineus on him. Now, cochineus and a bunch of other diatoms of this kind can move. They're plants that can move. And they move by secreting mucilage, which pushes them along. And they have a slit in them. But we are out of focus. Let me move him over just a wee bit. We'll go autofocus. And then slow scan. You can see that there's also crystals of salt. So the salt dried down as well and it crystallized. And if you've been in seawater, you know it tastes salty. Salt is sodium chloride, sodium and chlorine. And sodium chloride crystals are little cubes, but nobody told this one it was supposed to be a cube. And this one down here, let me just make him a bit bigger. Oops. This one down here is like a dodecahedron dice that the kids use on board games. So that tells you there's a lot more in seawater than just sodium and chlorine. There's magnesium and calcium and there's carbonates and sulfates and chlorides and then there's, there's so much in seawater. So the crystals aren't going to work as they would if it was a pure chemical mixture. Not mixture, this is a mixture. Okay, so now this guy is a baby. It's tiny and it's got the French fries and the marshmallows and the cocoa beans and also the biscottis and so on. But if we go over this way, you can see another one that's just a little bit bigger and it hangs by its feet onto the eelgrass and then it waves its top end around and catches stuff to eat. But on our sailboat, we took it out of the water because it needed cleaning the bottom. And there hanging was a whole bunch of weed. And of course I had to go pick it up and I could see a big one. So this is the head and its claws and things and its antenna. And so you can see this guy is really big. Whoops, I'm going the wrong way. Let's bring him over. And his front legs have got these claws to catch on to his prey. And then the back end starts to get spiky down his back. So I think he must be a different species. And he lost most of his tail. He got a bit crushed. And then it is the back end legs that hang on to the thing. But you can see the scale bar at the bottom says 1 millimeter. So from one end to the other end of a scale bar is 1 millimeter. So this guy's huge. You can see him. And the human eye has a resolution usually of about 100 to 200 micrometers. So if you take a millimeter and divide it by 1,000, you have a micrometer, and a bacterium is 1 to 5 micrometers. If you take the micrometer and divide it by 1,000, you have a nanometer, and a virus is about 30 nanometers. If you take the nanometer and divide it by 1,000, you have a picometer. Now we're in the atomic scale. So a silicon atom is about 235 picometers. And the resolution of our microscope down the corridor was measured at one point at 37 picometers. But then they put a repeater on the roof for cell phones, and we lost some resolution. But, you know, we got 50, 60 picometers. So you can see, you can see atoms. But did I mention atoms? They're all the same. Like you've seen one atom, you've seen them all. So anyway, so this amphipod has a common name, a skeleton shrimp. So if we go back, we'll go back to our, I'm not getting sidetracked by stopping on the way, because every time we get sidetracked, we find something new.
So Elaine, the skeleton shrimp you could see with your naked eye.
Yes, but the baby one would be difficult to see. I've lost him. Hold on a second. He's right by the Toblerones. Here we go. There. So he's very tiny. Well, let's have a look. If I take him up. So now the scale bar reads 500 micrometers. So 1,000 micrometers in a millimeter. So that's half a millimeter. Oh, wait a second though. You're in inches, aren't you? Wait a second now. Conversion. No, you're gonna have to work.
It's okay. It's okay.
You're
I
gonna
think
have to
people
work
know
in-
what a millimeter. A millimeter is small, very small.
Yes.
We've got centimeters on our yardsticks, so
That's right. On a ruler. Yeah. So, so this guy is about 500 micrometers across. So if we have a resolution of 100 to 200 micrometers, and that usually depends on how old you are, until you get glasses. And then once you get glasses, you get your resolution back. It's all very good. So we'll take him down. And we'll just go over here. And you can see that this is grayscale image. There's no color in a scanning electron microscope. If you find an electron micrograph that's colored, it's been colored in afterwards. Falsely colored afterwards. And you have to think of a rainbow. If you think of a rainbow, what's happened is the white light's been split up into all its different wavelengths. So the red end are the long wavelengths. And if you look at a rainbow, it goes from red through to blue. And blue are the short wavelengths. And the shorter the wavelength, the higher the resolution. But once you get past blue and indigo, then you're in the ultraviolet. And our eyes don't see ultraviolet because it's filtered out. So when they first started making artificial lenses, and they started replacing lenses with these artificial ones, the people that got them said, but I can see this other color. It's there. Well, would you ever try and explain to somebody what red looks like? To somebody who hasn't seen red before? It would be difficult. And these people would say, yeah, but it wasn't there before I got this new lens. And it turned out that our lenses filter the UV out because UV light is dangerous to the back of our eye. Some animals actually use it, like snakes or some insects will use it, but we don't. Oh, we got some tintinids and stuff on there. And we got a bare patch. Right, so I'm going up here. It's because we've used this so many times. Oh, what are you? See, I got sidetracked again. I just saw something. What is it? Oh, I think it may be an annelid. No, is it? Yes, it is. Look at that. There's an annelid. Let's go autofocus. And we can zoom in on that. Yeah. Hey, look at that. I didn't know it. And look at all these French fries. And the French fries have a scientific name, tabularia. Most of them are tabularia. But they're also the tines on a fork. The kids all say what they think they are. And then we kind of look for the biscottis, but I think these are the peanuts. And different species of cochineus. We've actually got about 46 species of diatoms on this sample. It's very cool. So, let's go back. I wasn't. She got side.
What was that in the middle there that was curled around? Did you say?
It's a worm.
Okay.
An annelid.
Another annelid worm.
Another annelid. Yeah. And you'll notice as well here. See these little black specks? These are tintinid mouths. So this tintinid over here, can we look inside the mouth? We'll have a look, try and see. Have a look at this one. He's actually not only covered, he's not only eating his food, he's covered in it. So he's got these, the biscottis in his mouth, these guys over here. Yep. And then, what about this guy? So you can see that there's a lot of tintinids on this thing. Oh, yummy. Yeah. Mind you, I'm not sure I'd like to eat diatoms because you've eaten all that glass. And this guy here's got filled up with them, with the tabularia, with the French fries. Whoops. Yeah. Mind you, you wouldn't have to go very far if you were covered up with your foods. You wouldn't have to worry about too much.
What is that little oval egg-shaped thing? Whoops.
Where?
Never mind. Oh, there it is. Over to the right of the mouth.
To the right of the mouth. Oh, there.
Yeah, that.
We'll have a look, because we can. Oh, cochineus. So it's another one of the cocoa beans.
Nice.
Yeah. So that's not a tabularia.
It's like tweezers.
Yeah. that's a different. Oops, that's a different species. Oh, and there's another one, another species, and there's another one. Ugh. And a lot of these you will be able to see with a good light microscope, because that's 50 micrometers. Yeah, you could probably see these guys with a good light microscope. Oh, and this one here is one of the triangular ones on a stalk. You can't see the stalk, and they grow as little colonies with stalks on the end. And so there's 5 species that we've got that look like this. There's another one, little fan-shaped ones, which are very cool. But I'm going to go and show you Bob. Bob's up here. The kids.
Elaine, I missed a couple questions here.
Oh.
I think this was with the tintinids, the ones with the mouths. How do they digest their food?
Don't know. That's another one. Now I expect there's somebody who would know that. I mean, I've just learned about tintinids. I've just learned the name tintinids because there's a lot of them on here. And once you see a lot of them, you kind of learn the name easily. But But this guy here was, yeah, so the more that we've seen how many there are on here, the more now I've got to look at the books. Yeah. So if the person does look in the books and finds out for me, they could email me and let me know. My email's easy, ech@uvic.ca. So here's Bob. He was named Bob by one class of kids, and he's been Bob ever since. But Bob is a copepod, and we'll go autofocus, and he's got short antenna. So we expect him to feed on other zooplankton, right? And if we zoom in some more, what we say to the kids is, you have your squidgy bits on the outside and your hard bits on the inside. But a copepod has his squidgy bits on the inside and his hard bits on the outside. So it'd be a bit like living in a cardboard box. You need holes in your cardboard box, or you need some means of getting oxygen in and out. And you need to be able to sense your surroundings. You need to know if you are going into cold or hot, up or down, fresh water, salt water. You need to be able to find your food, and you need to be able to find a mate. So you have a lot of sensory cells all over your body, and particularly the antenna. Now this guy got broken, but if we zoom in here, we'll take the reduced window allows me to focus on a particular part. You'll see he's got little sensory hairs all over his body, and I think I'm 98% sure, at least. I have a hypothesis that we haven't managed to actually figure it out, but I think these sensory hairs are in a pattern. So I think that they sense the way the water is moving over the body. It makes sense. If you ever look at a housefly under a microscope, you will see that the whole body, including the wings, are covered in hairs, and they are going to sense the movement of air around them. So when you go to slap a housefly, it doesn't see you coming as much as it feels you coming. So as soon as you get towards where it is, it's gone. You either have to go very, very fast or very, very slow. Now, you find copepods in pond water as well as seawater. You find diatoms in fresh water as well as seawater. And usually diatomaceous earth usually comes from an accumulation of a pond that's been filled in. And in Washington, DC, there's quite a few diatomaceous earth areas where they mine diatomaceous earth. And particularly the First Nations, and they called it just white clay that they used. But if we now move up here, here we have an ostracod. So the eelgrass has folded over, and we can see this guy here. So with a light microscope, you can see through this guy. Let's do an autofocus. And you can see his little legs, and you can see his heart beating in this one. So that's 200 micrometers. So he's about 150 micrometers. And there's some marshmallows. And there's one of the peanuts. And if we look a bit closer at the peanut, you can see it's covered in little diatoms. These little diatoms are called amphoras. So let me use the reduced window here. And it turns out, remember I said that some diatoms can move. And that's because they have a slit in the body that oozes out the mucilage. And this is one of the guys that's got this slit. You can't really see it very well here, but the slit is here. But ones that do produce mucilage tend to be very sticky. Well, you can imagine, right? And this background is the eelgrass. These are the cells of the eelgrass. But here, you can see how sticky it's got. These guys here are called Amphora. And this one, I think, is a navicula. Maybe another Amphora. But you can see the whole pattern on this is quite different from the whole pattern on the other ones that we've seen. The trouble with doing slow, it gives you a nicer image, but it does take a long time to actually come down, fill up the real estate. So the slit where the mucilage is coming is there. So a diatom is a bit like a petri dish. If I was in the UK, I would call it a petri dish. I had to learn a whole new language when I came to Canada. Let's see, aluminum and aluminium and things like that. And sometimes I forget. So I made a conscious effort to speak, to pronounce things the way my students would understand. And then very so often I forget. Was it vitamin or vitamin? That kind of thing. Anyway, so a petri dish, can you see this? You have to be having the speaker one. A petri dish will split and the top lid in a diatom grows a new one inside. The bottom one grows a new one inside. So the bottom one is going to get smaller and smaller and smaller, whereas the top one stays the same until the bottom one disappears. And then if there's enough around, they get sexual reproduction. There is a sexual reproduction phase, but mostly they do this asexual reproduction of splitting and growing new ones. But
so you're saying some replicate and get bigger and some replicate and get smaller?
Yeah, yes. The bottom cap gets smaller until it disappears. But the top one is going to stay the same because it's the top one that's the big one. But once they get sexual reproduction, then they go into a different phase of reproduction. But because it's a petri dish, let me go back here. The two sections of the petri dish have to be stuck together for the most part. And they have this rim around there that sticks the two together called a girdle band. And most women these days wouldn't know what a girdle is, but my grandmother used to wear a girdle and it was a very restraining piece of garment. Yes, I'm so glad I'm not in that age group that has had to wear girdles. No way, yes. Right, see, we get sidetracked so easily. Okay. How we
well, this is an exploration. I like the sidetracks.
Well, it's already 7:46, so we can go over. Oh, 1 minute. So there's a lot of caprellid amphipods and the like. Ooh, who are you? Oh, look at these guys, and these guys are called Ishmia. They're huge, huge diatoms here. This guy looks reasonable. We'll have a look at him. Well, it's because he's out of focus. Whoops. I got to get him in the middle so I can zoom in on him. There. And he's out of focus. So by now you'll know we have to hit the autofocus button. Actually, let's take the reduced window because I actually want it to be focused on here. Okay, so what does this remind you of? He's got more types of holes on him than anything. So the kids decided, oh, there's 8 in the chat. I hope we aren't missing anything. So the kids decided it was a bit like a microphone. But you can see, oh, he's got one of these long ones on him, a bit like a canoe. But the pattern on this part, let's just move him over. Whoops. Let's see, reduced window and drag it over. Autofocus. Oh, really? No, don't do that to me. That's better. There is a manual focus, but it takes a bit longer. So you can see the holes here are slightly different, but they have a pattern to them. Now this particular one hasn't lost as much of its organic coating as the Toblerones. And so the holes are covered in this very thin organic layer, and datum people tend to clean that off so they can tell what species they're looking at. But when you have an environmental sample that hasn't been cleaned off that way, it's only been sunshine that's cleaned this lot off, you can see that there's still a bit of goop on here. But this whole pattern is really, well, if you're a doodler, that would take you a bit of doodling, I have to say. So this is Ishmael. Huge, huge, huge. You could probably see that's 100. So he's at least 200, 250 micrometers long. So even an adult could see that one with the naked eye, just about. And there's a whole bunch of them here, all stuck together. Let's have a look at that lot. But and you can see how big this one is compared with the little tiny coconaceae. Oh, and there's one of, is that? See, I can get sidetracked very easily. This looks like, is this a round one on its side? Yeah, I think it is. So we take our reduced window because we want it in focus here. Oh yeah, it's got a lot of its organics still on it. So there's two here that are round, like a Frisbee. So they're very much like a Petri dish. And they're quite big too. So they're probably, now then, but there's two big round ones a lot off Galliano Island. And one is called Thalassocyra, and you'll have them on Vashon as definitely. Thalassocyra and Coscinodiscus. And this one looks like Coscinodiscus.
We had a question here from Carlista saying, how do you spell what is on now? Is it what we're seeing right now, or did I, Carlista, you can unmute and ask if you want.
Ishmia is Ishmia, and Coscinodiscus is CO. Coscinodiscus. It ends in DISCUS. Yeah.
Should let the kids name these. Lewis and Bob are a lot easier.
Oh yeah. It's also, what do they remind you of, right? Which is where we get the French, so the first lot of kids, and the kids come up with new names, new ones, and I have a list. Where's my list? Yeah. So I keep adding them to the list. So we have canoes, marshmallows, frisbees, matchsticks, cocoa beans, biscottis, peanuts, guitars, bananas. Oh, we have bananas. We didn't see the bananas. No. And the Kinder Surprises, fans, microphone, cushions and pillows. We didn't see cushions and pillows. Cushions and the pillows are different. And then the Diatomaceous Earth. Yeah. But we've seen the animals. So we are limited on our animals on here. Mostly because they would come in to feed on the things that are on the eelgrass. All right. Well, we've got time to do some more exploring on this. I know we're running out of time at the moment.
Yeah, let's do one. Take us one more place on this blade of eelgrass, and then I might want to open it up in case anybody has any questions or comments they want to give Elaine about this exploration.
Well, by now you'll be seeing all the same things. So now wait a minute, let's do a slow scan. Oh, there's a piece of a Coscinodiscus without its organic coating up in this corner. And there's one of the canoes. And there's some of these fan-shaped ones. a little piece over here, and there's, ooh,
And the bulk of what we're seeing here are all diatoms, correct?
yes, I only see diatoms and salt crystals.
Mhm.
I don't see anybody else, but these fan-shaped ones. So I'm working. We're working on an actual scientific project off Galiano Island through IMERSS, and we have UBC, IMERSS Lab, and me working on this. And I have a few volunteers who retired and wanted to volunteer because they got bored. So one of them was a medical doctor, and the other one worked at the IOS, Institute of Oceanographical Sciences. And they've come in, and one of them comes in on a Wednesday afternoon, and the other one comes in on Thursday afternoon. And we are looking at the species on particular blades of eelgrass. So we've taken second year growth and split it into the different proximal, distal, and medial. So the top end, the middle end, and the middle bit and the bottom end, because scientists have got to give it special names. And then we're identifying the species that live there. So wait a minute, I sidetracked where I was going in that. But if we can zoom in a bit. Oh,
Are you keeping track of quantity too? I mean, I just look at this and think, my God.
So when I see this, the first thing I tell them is, I'm not counting this one. That's what undergraduates are for. I don't mind counting the underside, which doesn't have as many on it. What we did worry that we were putting one side down and not seeing them all. So we had a look. We actually did an experiment to see which. We had it on a stick, and then we rotated it round so we could look at both sides. And there's one side that doesn't have as many as the other side. So I voted for counting the proximal backside of the eelgrass, and getting somebody else to count this lot. Yeah, I think bird people have the same problem when they have flocks of birds, and so there are lots of ways of doing it now. Yeah, that's. You want to open it up to questions?
Yeah, sure. So if anybody has any questions or comments on how your feelings when you're going through here, thinking of this as one little blade of eelgrass, I know it looks incredibly textured to me, and I've always thought of eelgrass as rather smooth, but having this up close look, I'm not gonna be able to look at eelgrass the same anymore. It's amazing.
No, you are not.
I think that it would be fine if anybody had questions, if you want to just unmute and ask. I think since I can't see people, that might be the easiest way to go.
Bianca?
Yeah.
Elaine, you sound or I sound like you do when you find something exciting. You're looking at, oh, wait a minute. Let's go back and find this. And what I enjoy, I do the HAB here for sound toxin on Vashon Island. When you look at a Cosendiscus and you start noticing the rays of the little circles.
Yep.
When not all of them are in rays, but as a knitter, you have to increase every so many rounds in order to have enough to make your circle get bigger.
That's right.
And it's always fun to me to look for that increased stitch on the Cosendiscus.
Oh yeah.
Oh, interesting.
This one here's got a lot of organics still on it.
Oh yes.
So the holes are covered up.
Well, and also it's not the same pattern you showed earlier, same kind.
So it's a different species.
Yeah.
There you go.
So Elaine, Carlista does plankton monitoring for sound toxins for I don't know how many years now, Carlista.
Oh,
So you guys would love each other. You could go out to
12.
tea and talk all day.
Okay, so when I come down, that's what we'll do.
It looks like Steve has a question. I see your hand up, Steve Bergman.
Yeah, thanks very much for these great images, Elaine. I'm just wondering about when these diatoms are living, what percent of their body is the skeleton that, the silica that we're seeing, and what percent is organics, the living cell stuff?
Well, plants tend to have a lot of vacuoles in them, right? Plant cells. So how much, now if we can find a broken one, you'll be able to see how thick the silica is. And it's not very thick.
Yeah.
See, there's a broken of the marshmallow kind. Oops, I've got to get in the middle. There we go.
Oh, wow.
You see that the silica coat isn't very big. Let's try an auto. My auto might not work. Oh yeah, there you go. Yeah, so really quite thin around the outside there.
So like humans have maybe 20% skeleton and 80% flesh, organics, right? And water.
Yeah, probably similar.
Okay, cool, thanks.
I would think, I'm guessing, but I know some diatoms are thinner than others. And we wrote a paper about cleaning diatoms, the different methods, because different labs have different methods. And some of the methods are really toxic because you use some really nasty chemicals and you boil sulfuric acid. You put them in sulfuric acid and you boil them. And others using, actually what Mark does very often is put a vial in his pocket and walks around with it for a few weeks. And he lets the bacteria feed on the organics. And then he's got nice clean diatoms without doing very much at all. But when we want something, we usually use hydrogen peroxide, but it doesn't work on all of them. And there's one that's in the Salish Sea called Ditalum. Actually, Ditalum is all over the world. And this particular person goes off to New Zealand or Indonesia or Germany or wherever, and he finds Ditalum. And it was very, very difficult to get a complete sample, unbroken, of Ditalum for the microscope, for the SEM, until we found a very, very delicate method of using a nuclear pore filter. You use a filter and you push the liquids over it. You don't mix them up and shake them up or anything because it just damages it. But Cassinodiscus and Thalassocyra, the frisbee types, they are very robust and they'll take just about anything you can throw at them and still come out unclean.
And the reason to clean them is to be able to classify them into different species,
right?
Because you can't see underneath the underneath structure.
That you can't see the holes and you can't see. So the slit, it's called a raphe slit, R-A-P-H-E, where the mucilage comes out. How that is arranged with the holes, the pattern, tells you the species. And a species can look the same, except one is a raphe slit that's the whole length, and one is split with a space in between it. And then another one will have one raphe slit on one side, but not on the other. And other ones have them on both sides. And suddenly species identification becomes a whole new game. And so unfortunately, I'm just the electron microscopist. And when we have a session and the experts come in, like you guys, I'm just using the microscope and they're coming in and saying, well, we need to just lift that up a bit. We need to tilt it a bit so we can see underneath. And then we find out that we can't actually see underneath. So we have to go find another one. But, one thing leads to another. Yeah. So
I think Fran Brooks in the comments says microorganisms are living on surfaces of nearly all creatures. A compare and contrast would be an interesting thing to do.
Yeah, well, that's really what we're doing. looking at this. The IMERSS group are really laying down the baseline. So we've just produced a paper of as many of the species that we can identify from 2 years ago, from a BioBlitz 2 years ago. And then every so often, we're going to do another big BioBlitz and look at Andrew. You remember Andrew, Bianca? Well, he wants to know all the species that lived on Galliano. And it grew from there. So when we have a BioBlitz, we usually have experts from all the universities, Royal BC Museum. There's a whole ton of people come in. So there's tree people and bird people, and there's the divers. There's a whole team of divers that doing the underwater stuff, and so on. And so this mini one was the lichens and all the things that we'd missed the first time round. But there's a paper coming out with all these species written down. So now in a few years' time, we can compare and we can see what's happening. There's a really good YouTube video of the dinosaur one. I think I sent it to you, Bianca, of the dinosaurs through the ages and how they changed and how they evolved, and then how they evolved into birds, basically. But he was saying that when the dinosaurs lived on the earth, we had no ice caps. So we were in a global climate that had no ice caps. And the land looked very different to what it is now. And he was looking at the dinosaurs, especially on the west coast of America, USA, Montana and Ohio, and in that area. And that whole thing was really good because his normal doesn't have ice caps. Our normal does, it seems. Now you see I've sidetracked totally where I was going.
Well, we have another question from Hazel that says, are you able to mark a point on a specimen so you can return to that place to study it?
Yes. So what we do, let me stop sharing this screen and share my screen on my computer. And we'll go into that PowerPoint again. So what we do is we take a blade like this. Let me just up this so it's bigger. And then we cut it up into different pieces, right? And then it goes onto a stub. So at one end, oh, what happened there? Come back. Yeah. At this end, the top one would be the one that I would volunteer to count. But the one underneath, not so much. But it's only a few centimeters. It's about a few it's quite narrow, and the length, the width of the eelgrass. So they all we looked at it as to how much we needed. So there's a 1 millimeter scale bar there. So this would be the length of the eelgrass across, but just a small piece of each area of the eelgrass. What was the question again?
whether you can mark a place and return to it. Yeah, so you can see this eelgrass has got all these French fries sticking out on the edge. There's tons of them. Oh my goodness. Yeah. So we've spent a few months looking at panoramas. This is a panorama of images taken on the SEM that we've put together in Photoshop. You have to do a good overlap to get it to see what was on there. So you could stitch them together. So this is a stitched one as well. Did we have any more on here? Yeah, so these are all cochineus, because cochineus is now a word that you know in your vocabulary. And there we have the microphone and the French fries
and the kids and the frisbees and the marshmallows.
And over here on the far left, it looks like a Cosendiscus on its side,
Here,
up higher, up. Yeah, that one.
no, that's Ishmael. I'm pretty sure that's
Okay, I've
Ishmael.
got to learn that one.
The microphone.
Yeah.
Yeah.
So it's
Wow.
kind of a buried Ishmael.
And then they come like this, with this folding in them. These ones here have this fold in it, and then this one doesn't. See, and this one is bent. It's got a fold in it. Oh, a canthan and an achanthia. Something. It starts off ACHTH. Achanth. No, there's an AN and a TH. There's a whole bunch of THs in there. This one here is called an amphora. I remember that one.
Yeah.
And then these long ones are tabularias. This one's a tabularia. Yeah. What else has I got on this slide? Oh, there we are. That's the end. Yes.
Okay, well,
Wow.
wow, thank you, Elaine. That's pretty amazing to be able to zoom in that far into life. Do you happen to have a piece of eelgrass laying about in your lab just to hold it up for people, just to give us some perspective of what we've been diving into, the size of the world?
I did. And I did have it right here, but I must have taken it down the corridor. Oh, right. Yeah, I took it down the corridor.
All right. Well, we can all find one sometime on our beach walk and remember this.
Oh, yeah. They're not, but the blades are not very wide. They're
Right.
just a few millimeters wide,
Mhm,
maybe 1/4 of an inch to 1/2 an inch.
mhm.
And but they're quite long. Yeah. So it's an ongoing process. But there's a ton of bacteria that are on them as well. And some filamentous bacteria that had us, we thought they were the hyphae of fungi, but now we think they're filamentous bacteria because they're very, very small. And the UBC group are part of the team because they're doing the DNA. And so they are going to tell us what species we have so that we haven't missed anything. But they're also very interested in the microbiota of the whole thing to see what is going on there. Because usually, you know, if you have a sailboat, particularly, the first thing that comes along are the bacteria that form the slime on the bottom of your boat. And that allows the barnacles to come along and attach.
Oh.
Yeah. See, barnacles really interesting because barnacles have a glue that works underwater. And in the old days, it used to, the dentists of the world were really interested in this glue. Because if you ever get to the dentist and had a filling or something, they had to dry the tooth off very carefully before they applied the glue. But mussels and barnacles have a glue that works underwater. Like, so cool. Now there's an artificial glue that they've made to do the same thing. So they're not so worried anymore.
Yeah.
Well, thank you, Elaine. Does anybody else have any last minute questions or comments they want to share before we wrap it up for the night?
Yeah, I'm glad I'm not that many dollars on hand to go out and get one of these microscopes because I would never get to the end.
Welcome to my world.
Yes, yeah, it's just,
Well, the next time we talk, I'll have to talk about the mini biblet stuff. So we've got bird's nest fungus and the
oh
slime
yeah,
and the petals, the pansy petals. So if I
So we've got Mike, you should get some commission from microscope people because Danielle says this was so cool. I want to get a microscope of some sort.
so this is pansy petals.
Wow.
So, I mean, pansies are so beautifully colored, but remember, you only get grayscale on a scanning electron microscope. I love living in the digital age because otherwise we wouldn't have scanning electron microscopes.
Yeah.
But this is a dried one, so these have been dried down. But if you were an artist, to draw that would take forever.
Yeah,
But isn't that beautiful?
gorgeous. One pansy, one little part of a pansy petal, that
Yeah.
is.
That's right. And if you look at a rose petals are very smooth. They don't have all these little projections on them. Weird. And then
Incredible.
the pansy pollen is quite different from anything else. And then the sword fern and the fern spores are so, and then, you know, oh yeah. So the next time we'll have to do a mini BioBlitz through a forest.
That would be very fun, Ellen. Elaine, that would
Maybe
be really
next
great.
year. Next year.
We'll invite you back for next year.
Great.
Through the forest. That sounds great.
Okay.
Thank you so much.
You're
Yes.
welcome.
Lovely. And thank
Thank
you
you.
everybody for coming.
Yeah, thanks so much,
Thanks, Elaine.
Elaine. Really enjoyed it. And
Okay.
Bianca, thank you for doing such a great job of hosting.
I usually have two goals in my PowerPoints. I usually have two goals that I start off with. And I should actually show you what my two goals are. If I can open, is it this one? I think so. Yeah. So am I sharing?
You're not sharing right now.
Okay, let me just share my screen. Let's go Zoom. And I'll show you the two goals that I have. Share screen, this one. So here they are. And it usually comes up like this. The first goal is no one is to go to sleep. And the second goal is I want at least one wow from every member of the audience. Fortunately, with electron microscopy, that's usually not so difficult.
You did it.
Yeah.
I think you did you reached both of your goals.
Oh, good.
Well done.
Bianca,
Mhm.
I had one thought when you said something about interesting, are these microscopic animals, yeah, because they eat something on other surfaces? Think about the human being, the human body, just the face. Do we really want to look at that?
Yes. Oh, so I had a second class today of grade 12s, and they were doing human anatomy. And I have a stub with bits of mouse bits on it. And mouse bits, cilia and microvilli and blood cells and blood vessels are very similar. You can look at them, they're smaller in a mouse, but on an electron microscope, if you don't look at the scale bar, you just see the same thing. The tongue was different. Wait a minute, I'm now sidetracked off where I was going on that one. Yes, no, but the patterning under electron microscope, again, it can be so beautiful when you start looking at it very, very closely.
Yeah.
You see it in grayscale, so you don't see blood, you don't see red unless you color it in. The red color tends to make it a bit icky, but
We'll stick to pansy petals.
yay.
Roses.
Okay.
All right.
Thank you.
And thank you, Joe, for hosting, and Vashon Center for the Arts. It's a great partnership that allows us to bring these talks. So thanks very much. And thanks,
Absolutely.
Elaine. It was so great to see you again.
And you too. Yeah. Hopefully we'll see you in August.